Hello, everyone. Today, I would like to interpret the research results published by the research group of Shukang/Wang Chunli of Hubei University of Chinese Medicine on iMeta. Gut microbiota-derived butyric acid regulates calcific aortic valve disease pathogenesis by modulating GAPDH lactylation and butyrylation. First of all, I would like to briefly introduce the research background of this study. Calcific aortic valvular disease, CAVD, is a complex chronic pathological process. At present, there is no effective drug to delay its progression. A large number of studies have shown that intestinal flora as a key regulator plays a key role in the occurrence and development of cardiovascular disease. This study mainly explored the molecular mechanism of gut microbiota-derived metabolites involved in the occurrence and development of CAVD. In this study, multiomic sequencing and correlation analysis identified the beneficial role of prausnitzii-derived butyric acid, BA, as a key metabolite in the restoration of valve function in calcific aortic valve disease, CAVD. The therapeutic efficacy of BA in attenuating CAVD progression was confirmed in vitro, ex vivo, and in vivo. Subsequent mechanistic investigations revealed that BA reshaped glycolysis through site-specific inhibition of lactylation at the Lys263 residue of GAPDH, which is mediated by competitive inhibition of butyrylation at the same site. The following are the main findings. First, based on neural network algorithm and clustering analysis, we divided APO mice fed with high-fat diet into easy calcification group and difficult calcification group according to the cross-valve pressure difference and valve thickness. The visceral cage test showed that the calcification degree of mice in the dirty group was significantly reduced. The fecal transplantation experiment successfully reproduced the protective effect of the dirty cage environment and effectively improved the degree of calcification in mice. This indicates that the intestinal flora is closely related to aortic valve calcification. 16S rRNA sequencing analysis revealed that fecal microbial diversity was higher in calcified mice. Clostridium praecox was negatively correlated with the degree of calcification. The intervention of prausnitzii significantly reduced the aortic valve calcification area and BMP2 expression. At the same time, the supernatant of prausnitzii culture can significantly reduce the calcification of human valve tissue in vitro. The above results indicate that prausnitzii has anti-calcification protective effect, or can be used as a potential probiotic preparation to prevent cardiac valve calcification. Metabolomic analysis of mouse feces and serum showed that the differential metabolites were mainly enriched in lipids and lipid molecules, and butyrate was the metabolite with the strongest correlation with the abundance of prausnitzii. The level of butyrate in the difficult calcification group was significantly higher than that in the easy calcification group, and it was positively correlated with the abundance of prausnitzii in stool and serum. Exogenous supplementation of 13C labeled butyrate could significantly increase the concentration of butyrate in mouse heart. These findings confirm that butyrate can cross the intestinal barrier and enter the blood circulation, and play an anti-CAVD protective role in cardiac tissue accumulation. To clarify the protective effect of butyrate on aortic valve calcification, the research team carried out systematic in vitro and in vivo validation. 500 μm butyrate treatment significantly reduced calcium salt deposition in VIX induced by osteogenic induction medium and the expression of RUNX2 and BMP2. Transcriptomics and extracellular acidification rate detection experiments showed that butyrate could significantly change the glycolytic pathway of VIX treated with OM taken together. Butyric acid effectively reduces aortic valve calcification both in vivo and ex vivo in HVICs. Butyrate can inhibit the three lactylation sites of GAPDH, K194, K215 and K263, in VIX induced by osteogenesis. The site mutation experiment showed that the three site mutants significantly inhibited the calcification of VIX, and the K263R mutation had the most significant effect. Further experiments showed that butyric acid treatment significantly increased the butyrylation level of GAPDH K263 site, while the lactylation level simultaneously decreased. These results suggest that butyrate inhibits the activation of osteogenic signaling pathway by competitively promoting the butyrylation of GAPDH K263 site and blocking the calcification promoting lactylation. In conclusion, we identified prausnitzii, a key gut microbiota, as being negatively associated with CAVD risk. Prausnitzii slows the osteogenic differentiation of HVICs and decelerates the progression of CAVD through the previously undescribed metabolite butyric acid/epigenetic reshaping/glycolysis pathway. Furthermore, the present study demonstrated that prausnitzii and butyric acid are promising candidates for CAVD prevention and treatment.